it in a burst, yeah, right at FuSha Works.
Drug repurposing, breathing new life into old drugs. What is drug repurposing? Drug repurposing is a strategy for identifying new therapeutic uses for approved or investigational drugs beyond the scope of the original medical indication. Drug repurposing or repositioning is a technique whereby existing drugs are used to treat emerging and challenging diseases. Drug repurposing has become a promising approach as it offers many advantages of drug repositioning. It is a universal strategy for neglected diseases due to a variety of reasons. Firstly, it reduces the number of required clinical steps, which thus could reduce the time and costs needed for the medicine or drug to reach the market. Existing pharmaceutical chains and big pharma companies, such as GlaxoSmithKline, are able to facilitate formulation and distribution of the drug. Possibility of incorporation with other medications allow for combination drug therapy to improve clinical efficacy of treatment. Repositioning could allow and facilitate the discovery of new mechanisms of action for old drugs and new classes of medications. Rapid advancement into disease-oriented research also occurs due to the removal of barriers faced in early research stages. This list of advantages allow for drug repurposing to be a time and cost-effective strategy for the treatment of diseases with minimal risk of failure. Thus, drug repurposing maximizes the therapeutic value of a drug and subsequently increases its success rate. However, there are some drawbacks of drug repurposing. More often than not, there are different dosage requirements in treating a new novel disease as compared to the original target disease that this drug was formulated for. In such cases, the discovery team will have to begin again from phase 1 clinical trials and test out the new dosage requirements accordingly. This effectively strips away the main advantages of drug repositioning over discovering a drug from scratch. Secondly, there may be a need to find new formulation and distribution mechanisms of existing drugs in context of the novel disease in question. This may often differ from the intended biological target of the original disease the drug was formulated for. There could also be a patent right issue that may pose a stumbling block in drug repurposing. This could complicate issues as legal disclosure is required before scientists can move ahead in repurposing a drug. However, despite the disadvantages, the benefits of drug repurposing greatly outweighs the costs, and thus, it is an effective alternative approach to the traditional drug discovery process. To better visualize the time and costs associated with drug discovery, here's a flowchart showing the timeline for a traditional drug development process, as well as the timeline for a repurposed drug. Timeline A, which represents traditional drug development, involves a lengthy drug development process as well as clinical trials. Billions of dollars are needed to invest in research and development, and there is a high failure rate, with as many as 90% of drug candidates ending in futility. Timeline B, on the other hand, expedites this process greatly, especially so in the discovery phase. This allows pharmaceutical companies to market existing drugs faster and allows it to reach consumers within a shorter time span. Next, let's move on to some examples of common drugs that have been repurposed. Firstly, we have sildenafil, more commonly known as Viagra. Viagra was originally developed in the 1990s by Pfizer to treat hypertension and angina. However, it became apparent during clinical trials that Viagra induces penile erections, and the rest, as they say, is history. Another commonly known drug that has been repurposed is aspirin. This over-the-counter medication has been traditionally used for the treatment of pain, fever, or inflammation. More recently, aspirin has also been used for the treatment and prevention of atherosclerotic diseases. An interesting fact to note is that for this purpose, the aspirin tablet is left to dissolve under the tongue rather than swallowing it whole. This route of administration is also known as sublingual, Latin for under the tongue. There are two main strategies that eventually result in the repurposing of a drug. They are known as on-target drug repurposing as well as off-target drug repurposing. As with all drugs in the market, the main target on which they act on is already known. Together with the vast clinical trials and patient data available, drugs may be found to have other side effects or indications. This leads to the first strategy, known as on-target repurposing, as there's repositioning around a known drug target. Apart from that, a drug may also be further investigated for its effect on other biological targets, usually via high-throughput screenings. In this off-target repurposing strategy, a drug may be found to have an effect on a new target, which gives rise to a new therapeutic indication instead. Let's take a look into these two strategies in more detail. The on-target strategy takes advantage of the already known pharmacological mechanism of a drug. Though it has the same biological target, it can be repurposed to treat a different clinical disease from what the drug was originally intended to treat. An example would be minoxidil. Minoxidil was repurposed from an antihypertensive vasodilator to an anti-hair loss drug. Minoxidil's mechanism of action serves in widening of blood vessels and opening of potassium channels. Thus, it makes sense that it's used as a vasodilator. However, it has now been repurposed as an anti-hair loss drug because vasodilation also allows more oxygen-rich blood with nutrients to reach hair follicles, thus promoting hair growth. The off-target strategy, on the other hand, stems from a previously undiscovered mechanism of action. This newly discovered mechanism of action would then provide new targets for existing drugs. An example would be aspirin. Aspirin was repurposed before, and now it has been repurposed once more. As mentioned previously, aspirin was used for atherosclerotic diseases as it suppresses blood coagulation, but a new mechanism of action was discovered that allows it to be repurposed for the treatment of prostate cancer. In either of the two drug repurposing strategies, on-target and off-target, screening must be done. This is done to discover either new biological targets with the same mechanism of action or new biological targets with a different mechanism of action. Screening can be done via two methods. Firstly, either experimentally or activity-based through experiments, or secondly, it can be done virtually, which is also known as the in silico approach. Let's take a look into these two methods in more detail. An experimental or activity-based screen involves screening of existing drugs for new pharmacological indications based on protein target and cell-based assays. The use of various in vitro assays and in vivo models can help to expedite the screening process. Since this requires no information about the target, it can be done relatively easily, but it will, however, be a time and labor-intensive process. On the other hand, a virtual-based or in silico screen takes advantage of known existing databases and uses computational biology and bioinformatic tools to enable a high throughput screen of the databases. The fact that we are using a database also means that we already have information about the target. It is often a more efficient method of choice, but it should be noted that it also results in a higher rate of false positives. Thus, we can use a combination of both experimental and in silico approaches to experimentally verify the positive results obtained from the virtual screen. Moving on, let's take a look at a specific case study: repurposing peptide toxin as anti-infectives. This paper was recently published in the Proceedings of the National Academy of Sciences in 2020. In nature, there are many species of flora and fauna which produce their own peptide toxins. These species include wasps, snakes, spiders, and even the sea anemone. What is fascinating is that these toxins possess some anti-infective properties, such as antimicrobial and anti-cancer properties. In this case study, the authors focus on Mastoperin L, or MAST-L for short. MAST-L is a peptide toxin derived from wasps, and it possesses some antimicrobial properties. These antimicrobial peptides are a promising class of antibiotic candidates, as they have an almost unlimited sequence space, coupled with multiple mechanisms of action against microbes. Another advantage of these antimicrobial peptides would be that of antibiotic resistance. Bacteria exposed to antimicrobial peptides develop resistance at a much slower rate as compared to conventional antibiotics. However, due to MAST-L's toxic profile that targets the human cell membrane, it cannot be used as a potential therapeutic without first eliminating its toxicity. Initially, the researchers performed an in silico screen using the extensive antimicrobial peptide database and identified a unique pentapeptide motif, which is a 5-membered amino acid sequence known as phenylalanine, leucine, proline, isoleucine, isoleucine, abbreviated as FLPII. This pentapeptide motif was observed to be conserved in the N-terminus extremity of peptides with high antimicrobial and immunomodulatory properties. Thus, it makes rational sense to create a synthetic peptide that incorporates the pentapeptide motif into the N-terminus extremity of MAST-L. It should be noted that in this case study, the mechanism of action and biological target remains unchanged, and modifications are made instead to improve its efficacy. After the insertion of the conserved pentapeptide motif, which did not alter the helical structure of the peptide, a series of in vitro bacterial growth inhibition assays were carried out to evaluate if the motif influenced the biological function of the peptide. Here, the minimal inhibitory concentrations of the peptide against 15 different pathogens were assessed. Overall, the new MAST-MO is slightly superior to its predecessor MAST-L, and is also comparable in activity to standard antibiotics. Briefly, MAST-MO also presented an increased activity against gram-positive bacteria over gram-negative bacteria. The only exception of lower activity against gram-positive bacteria was that of MRSA. Following which, in vitro cell line toxicity studies were conducted for MAST-L. In particular, the effect of the motif insertion into the peptide on its subsequent cytotoxicity was evaluated. Preliminary in vitro studies revealed that no hemotoxicity was present, but it was noted that MAST-MO is toxic towards the human embryonic cell line HEK293. In comparison, the previous MAST-L was both hemotoxic and toxic towards different human and murine cell lines. Thus, in order to deplete the remaining toxicity observed in masked MO towards the human embryonic cell line, a permutation library of 59 masked MO derivatives, which composed of single amino acid permutations for the FLP-II motif, were assessed on both antimicrobial and cytotoxic activities. Two analogs, analog 15 and analog 49, did not show any side effects or adverse toxicity, while also displaying increased antimicrobial activity compared to the parent masked MO. Thus, these analogs of masked MO can be used as an active ingredient to derive future pharmaceutical drugs. In summary, multi-drug resistant pathogens are becoming increasingly prevalent, and no new classes of antibiotics have been discovered for the past decades. Strategies such as drug repurposing can be leveraged on to create a new class of drugs or antibiotics to treat bacterial infections. The case study discussed also shows that toxins from the venom of insects display antimicrobial properties, and it represents how nature can be an untapped source of template molecules for the design of future novel drug candidates.